Good morning, everyone. Welcome to today's MPG primer. We're hybrid today, both in person with a delicious breakfast as well as remote. So if you're on your way in, please feel free to join us at any time in person. I'm very excited for today's talk. We will get to hear from Dr. Sean Misek, who is a postdoctoral associate at Dana-Farber Institute and the Broad Institute. He earned his PhD in physiology at Michigan State University, and his postdoctoral work now seeks to understand how germline variation shapes cancer vulnerabilities. And at present, he is focused on genetic drivers of low-grade glioma growth. And we're very excited to hear his talk today, which is a little to the side of that main talk, but applicable to so many people's research in many areas, which is looking at germline variation as it might contribute to false negatives in CRISPR-based experiments. So Sean, thank you so much for joining us today. He's graciously happy to take questions during the talk. And so please write in the Q&A or raise your hand or come to the mics in person and he'll take your questions as he goes. Thank you.
Yeah, so I've watched a few of these videos, on YouTube, which has been great. Although this is, my first one in person, so that's exciting too. And I'll definitely come to more in person now that they're, no longer virtual. Yeah, so I guess the story I'm gonna tell today is maybe a little bit about how I was originally excited about a lot of cool data and then realized that I was being led astray. And so like the river on this slide is just a metaphor for how the path to getting here was very windy, but somehow we found our way. And so one disclaimer is that I'm not actually a population geneticist. And so as a form of appeasement, I've decided to put a Manhattan plot on slide 1. And so I'm generally interested in cancer genetics, like many people here at the Broad. And I think one really powerful thing about cancer genetics so far is that people have been able to identify a whole bunch of genetic variants that are associated with risk of developing cancer. And so this is just the Manhattan plot showing overall risk of developing cancer that's diagnosed by a doctor, and it's looking for genetic loci that are associated with that. And what you can see is that there are really a few genes that stand out above the rest. And in particular, there are these two genes, BRCA1 and BRCA2. And I think the cool thing about these two genes is that individuals who have these genotypes can elect to undergo preventative measures in the clinic to reduce their risk of developing cancer. And so, identifying these genetic variants has really been life-changing for some cancer patients. But the thing that I'm really interested in is this idea that your own genetic variation can really engender specific cancer vulnerabilities in your tumor. And I'll kind of get back to this a little bit at the end, but I feel that this idea has generally not been explored very thoroughly. But one example of where this has been very powerful is again with these same BRCA mutations. And so the idea here is that if your tumor has 2 wild-type copies of BRCA and you treat that tumor with a PARP inhibitor, because these tumors are still proficient in DNA repair, these tumors can really shrug off this treatment and they're not very responsive. But in a subset of patients, they have germline loss of 1 BRCA allele, and you get somatic loss of a second BRCA allele in the tumor. And because the tumors no longer have any copies of this BRCA gene that are functional, when you treat these tumors with PARP inhibitors, because they're deficient in DNA repair, they're exquisitely sensitive. And so I think this concept of, again, germline variants engendering cancer vulnerabilities is, unexplored relative to the topic on the first slide. And I think it's really interesting. I think this plot here really kind of shows why I think this is interesting. And so what this plot is it's just looking at cancer survival rates in the US across a range of cancer types. And it's stratified based upon genetic ancestry. So European ancestry in blue and African ancestry in orange. And the take-home point for this plot is that cancer outcomes in the United States differ greatly depending on one's genetic ancestry. And so, what this plot really shows is that the survival rates for individuals of African ancestry today is about the same as the survival rate for individuals of European ancestry around the time that the human genome was being sequenced. And so there's this huge cancer disparity. And I think it really shows that, genetics in kind of dictating cancer outcomes is potentially a important problem to work on. And so this is actually a very complex problem. And certainly there are large socioeconomic contributions to this. But we think that there's also a genetic component to this as well. So we know for sure genetic variation differs across ancestry groups. We also know from recent work in our lab and others that there are different patterns of somatic alterations within tumors from patients with different ancestry groups as well. And so what we want to know is whether these differences in germline and somatic alterations translate into differences in cancer vulnerabilities, and whether this is also contributing to disparities in cancer outcomes. And so the main question we're really trying to answer, at least initially with this analysis, is are there cancer vulnerabilities that differ across ancestry groups? And so how do we go about answering that question? I'm not a population geneticist, as I said, I'm a cancer biologist. And so we turned to the Cancer Dependency Map. And if you're not familiar with the Cancer Dependency Map, what it is this large profiling effort at the Broad for a range of cancer cell models across a range of different lineages. So to date, around 1,000 cancer cell lines have been extensively profiled on the molecular level, looking at RNA expression and DNA phase readouts. And they've also been functionally profiled as well with chemical compounds, drugs, and important for the analysis we did, they've also undergone genome scale CRISPR screens to systematically identify genes that are required for the survival of these cells. And the power of this data cube is that now you can begin to build models where you can associate specific genetic features in these cell lines with cancer vulnerabilities. And so what we can do is we can use this question to ask, does the genetic ancestry of the cell line, is that associated with any cancer vulnerability? And so how do we actually figure out what the genetic ancestry of the cancer cell line is? So we can do much like many do in the population genetics field, and we can take this a known reference where we have a bunch of genotyping data from patients of known ancestry groups from 1,000 genomes. And we use this algorithm called RF mix, which will allow us to infer ancestry at each specific locus in the genome. The reason why we want to do this is because we'd like to be able to say, okay, in this cell line, the region of the genome that has the EGFR gene is East Asian ancestry, and this region of the genome that has another gene in it is of a different ancestry group. And so it gives us more power to kind of really detect some of these differences. And so we did this, and we were actually very surprised that the results were not super good. Actually, it's actually quite disappointing because when we looked at all of the cell lines in DepMap, which represent a majority of the cell lines that we use in basic cancer research, an overwhelming majority of them were either of European ancestry or East Asian ancestry, and only a minority of these cell lines were of African or South Asian or American ancestry groups. And so what this kind of suggests is that when we do experiments in the lab to test some new hypothesis that a gene is important for cancer, or maybe do preclinical testing for this compound that's going to go into clinic, we're almost always just testing it exclusively on European and East Asian cell lines. And so already, like, certainly this is a problem for the analysis we want to do, but it's a problem for research in general. And so we thought, okay, we can't really detect these differences in all the ancestry groups like we originally wanted to do, but can we detect differences between European and East Asian cell lines at least? And so just to kind of explicitly show what we're doing here is now we have a list of ancestry in all of these cancer cell lines that we're looking at. And we know in each cell line which genes are required for their growth and which genes are not. And so we can begin to make these comparisons and try to identify ancestry-associated cancer vulnerabilities. And so just to kind of like orient you for how we're thinking about these cancer vulnerabilities. So in DepMap, the metric for a cancer vulnerability killing or not killing a cell, we call it just a Kronos score, which is an amalgamation of all the different guide RNAs into a single gene. And so this here is just an example of a distribution of Kronos scores for the EGFR gene, where it's a distribution of cell lines. And you can see that a majority of them cluster around 0, which means that if you knock out EGFR in these cell lines, they're largely not affected in growth. And you can see that there's a subset of cell lines, there's this long tail where it goes out to -1, which indicates that these cell lines are very dependent on EGFR for their survival. And a median of 0 for the Kronos score is just the median of non-essential genes across all cells. And then a Kronos score of -1 is the median of all common essential genes that we think are generally required for the survival of cancer cells. And so a difference of -1 is quite substantial. And so when we did this analysis, what we found is that surprisingly, we were actually able to identify quite a few genetic dependencies that are associated with cell line ancestry. Unsurprising to us, a majority of them are in either European cell lines are associated with European ancestry in blue or East Asian ancestry in green. And it's not surprising because a majority of the cell lines in our study are of these two ancestry groups. But importantly, we were able to identify some associations with African ancestry as well. And so this is really promising, and we really wanted to kind of understand the nature of why these genes are associated with genetic ancestry. And so initially looking at this kind of volcano plot, I was looking at all the genes, and I actually didn't really know what many of them do. I don't think I've ever heard of any of these genes before, but when we were looking through the data a little bit more, we found 2 examples of genes that we were initially quite excited about. One of them is PTPN11, which is a stronger dependency in East Asian cell lines compared to European cell lines. And the other is GRB2, which has the same pattern. And one reason why we were initially very excited about this is because we know that EGFR mutations are more common in East Asian lung cancer patients compared to lung cancer patients with European ancestry. And we know that GRB2 and PTPN11 are essential for EGFR to signal downstream to induce proliferation. And so, we already see these sorts of associations that kind of make sense in our head. And what we really wanted to do is understand why are these dependencies selective for East Asian over European, and we really wanted to understand it on a molecular level. And so since we know that one of the defining features of genetic ancestry is that there's differences in patterns of germline variation across ancestry groups, we next wanted to look genome-wide to try to identify loci that are associated with these dependency profiles. And so we did this for all of the ancestry-associated genes we found, and this is just one example here. And what we found is that in many of the cases, we identified these very significant loci that are all proximal to the ancestry-associated dependence in interest. So, for example, here in this Manhattan plot, we're looking for genetic loci that are associated with dependence on this gene called claspin. And all these variants are proximal to the claspin gene. And when we look more globally at that analysis on the right, what we can see is that generally cis PTLs are much stronger signals than some of the trans PTLs. And so we became interested in trying to understand these cis PTLs in particular. The first thing we thought is, well, maybe since a lot of these are around the gene or even in the first few exons of the gene themselves, maybe they're helping control expression of the gene, and that's why we're seeing these differences. And so we did this analysis, and we took all of these variants, and we attempted to identify if these are eQTLs. And we actually found quite a few instances where there are. So in about 20 of the 98 ancestry-associated genes, the most significant SNP is actually associated with expression of that gene, which I think is really cool. But in a majority of these cases, what we found is that these variants are not associated with expression. And what's even more concerning is that when we just looked at expression of these ancestry-associated genes across all the cell lines in DepMap, what we found is that a majority of them are not even expressed at all. And so we're knocking out genes that the cells are not expressing, and somehow it's eliciting this phenotype where the cells are dying. And so I think it's safe to say that at this point I was getting a little concerned about the data, and we're immediately starting to think that, well, maybe there's a bug in the code, or there's some sort of artifact. And so one way in which this artifact could potentially manifest is that we know that differences in DNA cuts, like the number of DNA cuts, can potentially be cytotoxic. And so what I mean by that is, for example, we could have European cell lines where they have copy number amplification at some locus, and East Asian cell lines where they don't. And when you introduce CRISPR to these cells, you're going to cut at all of these genetic loci. And so in the European cell lines, you're going to have 5 cuts, and in the East Asian cell lines, you're going to have 1 or 2 cuts. And so the idea here is that because you're getting more cuts in the European cell lines, it's going to be more toxic because you're inducing more DNA breaks, and so the cells will just die because of that. But this has already been corrected for in the cancer dependency map, so we know it's probably not due to copy number. And so instead we looked in the other direction and we said, well, is there anything that would prevent CRISPR from cutting? And so one way that CRISPR could be blocked from cutting is that you could have theoretically cells without a SNP on the left, and you get a perfect match between the CRISPR guide and the reference genome. And you can have cells with a SNP in the guide targeting sequence on the right, which could preclude guide binding. And so what ends up happening is if you have a perfect match, you get DNA editing and you get a cut and a double strand break, which is potentially cytotoxic. And on the right, because that SNP is introducing a mismatch between the guide and the genome, you won't get cutting, and so you won't have that cytotoxic effect. And consistent with this idea, when we mapped all of the variants in cell lines to guides targeting these ancestry-associated genes, and so each row in this heat map is, you can think about it like a guide that's targeting an ancestry-associated gene, and each column is a different position within that guide. What we found is that, again, a majority of these guides that are targeting ancestry-associated genes actually have SNPs that map to these targeting sequences. And this is much more than we would expect by random cans. And so what this kind of suggests is that there's this genetic variation that's specific to different ancestry groups that's blocking CRISPR from cutting in different patterns across different ancestry groups. And just kind of an illustrative example of that. So we're looking at, on the left, 4 different guides that are targeting this claspin gene. And so you can see that for the first 3, the guides are equally effective in European and East Asian cell lines. But then you can see for this last guide, the guide is able to cut and kill European cell lines a little bit, but it's not effective in East Asian cell lines. And when we look, what we saw is that there's a SNP that's very prevalent in your East Asian cell lines in claspin guide number 4, but not in any of the other claspin guides. And we can look at this in a different way across all of the guides in DepMap, and we can say, how does the position of these mismatches in the guide influence its cutting activity? And so excitingly, what we found with the box plot part of it is that generally cutting activity is more affected as you get closer and closer to the PAM site. And this is consistent with other data from John Dench's group showing the exact same thing. And so it's exciting that we're able to detect that using a completely different method. And so then what we did is we just looked, again, genome-wide, not focusing on the ancestry-associated genes, just to figure out how much of a problem is this in DepMap in general. And what we found is that there's about 800 different genes where there's an association between a germline variant in at least one of the guides and the overall gene dependency score for those genes. And we found 47 of them where this relationship is very strong. And so I think this really kind of highlights this problem where, because we're not accounting for genetic variation, especially not accounting for genetic variation across ancestry groups, we're sort of introducing this bias into all CRISPR-based experiments, not just those in DepMap, where maybe if you do a CRISPR experiment in, a cell line of East Asian ancestry, you could be misinterpreting your data as a gene not being essential for growth, when in reality, your CRISPR reagent is just not working. And I wanted to highlight this paper as well that was published from Luca Pinella's group just a few months ago, where they found the exact same thing, which is really cool, but the opposite. And what they found in this paper is that you can actually have germline variations that introduce new CRISPR cut sites into the genome, and there's potential therapeutic relevance for these as well. And so it's a really cool paper. And so what we really wanted to do now is we wanted to understand how does this artifact affect different ancestry groups and different populations. And just in general, if you have a cell line of some ancestry group, how many guides in your library or how many CRISPR cut sites are potentially affected? And so what we did is we took all of the variants from the 1,000 genomes and HTTP subset of NOMAD, and we mapped them to guide targeting sequences from many common CRISPR libraries. So at the top, what we did is we just found 1 million random guide sites throughout the genome, and we mapped SNPs to all these sites. And what we found is that on average, about 2% of these sites in the genome were affected by this artifact in each individual. So if you did, if you screened 1,000 guides in a cell line, 2% of them would not work. But what was shocking to us actually is that African individuals or individuals of African descent were seemingly more affected by this artifact than individuals of any other ancestry group. And so we wanted to know, is this just maybe an artifact of how we're picking these random guides, or is this actually manifesting in these CRISPR-based experiments that we're doing? And so we looked at many of the common CRISPR libraries that we use at the Broad and other folks use at other institutions. And we see this exact same pattern where the fraction of guides that are affected in each library differs based upon how the library was designed. But the consistently individuals of African descent appear to be more affected by this artifact than individuals of other ancestry groups. We also looked at CRISPR libraries that were designed to avoid regions with high variation without necessarily paying attention to whether that variation is specific to different ancestry groups. And what you can see on the x-axis is that the rate at which these guides are affected is much lower, but it still retains this bias at the same ratio where individuals of African descent are consistently more affected by this artifact than individuals of other ancestry groups. And so, we wanted to really try to do something about this problem. We wanted to see if we could design a CRISPR library that removes this ancestry bias. And so what Aaron O'Connor in the lab did is he built some rules to try to design guides that are equally effective across ancestry groups. And so what you can see here in this bottom plot is that now once you apply these rules, the guide design, you can see that the rate at which guides are affected is the same between individuals of African descent and individuals of other ancestry groups. And he named this library, I can't pronounce it, but this is a grape cultivar that's popular in Morocco. And the reason he picked that is because all of these other grape cultivars that the Broad libraries are named after grapes that are popular in European countries. Okay, and so now we're kind of changing the focus and we're trying to, we've identified the problem. And so how can we actually solve this problem? And so we really took 3 approaches to try to solve it. The first is we want to make sure that when people design CRISPR guides, they're not designing them to target regions with high genomic variability. And so we're working with the PTP team at the Broad to help incorporate new data into their CRISPR guide design algorithm. And so the idea is that we can feed in genetic variation from NOMAD, and we can help avoid regions that have high genetic variability. And so, hopefully at some point in the not so distant future, when you go to design guides for your own experiment, using this tool, you'll hopefully avoid this issue. Aaron and Leah on the pattern team also built this really cool web interface. And so what you can do here is if you have your own guides for your own experiment, you can just plug them in here and you can figure out if your guide, like how effective is it by genetic variation across ancestry groups. And you can also browse guides from common CRISPR libraries as well using this tool. So it helps you just design better guides and make sure that when you're designing guides for your own experiments, you're avoiding regions of high variability. And then finally, we also wanted to help solve this problem in the Cancer Dependency Map and really kind of show potentially how to solve this problem with CRISPR screening in general. And so we worked with the DepMap team, and what they came up with is this idea where what you can do is you can identify in each cell line which guides have a SNP in their targeting sequence, and then you can just remove these from the gene level dependency summary. And the idea is you can just recalculate these scores based upon that. And so this has already been incorporated into DepMap. So if you're using DepMap data, the current release has this integrated already. And so what we did is we went back to that original analysis we did, or original question that we asked like forever ago. We asked, okay, is this bias that we identified actually removed in the data? And so what you can see here is that the x-axis is before correction, and you can see that there's a whole bunch of significant associations down here. And then the y-axis is after correction, and you can see that there's really not that many that emerge only in this new data set compared to the old version of the data set. But what's exciting to us is that we still see towards our original question, we can identify a whole bunch of associations that are significant in both data sets. And so what this means is that these associations in the top right quadrant are not due to this SNP artifact, and they're potentially real or due to some other artifact that we haven't found yet. But excitingly, the two examples that I showed at the very beginning, PTP11 and GRB2, that we were very excited about, they still hold true after we correct for this SNP and guide artifact as well. So they're probably due to some other artifact that we just haven't found yet. And so moving forward, we're really interested in kind of really understanding these ancestry-associated dependencies. And just more broadly, we're interested in understanding how individual germline variants may engender cancer-specific vulnerabilities. And so I have really three main points that I'd like to hammer home. So if you're sleeping, wake up. So the first is that the cancer models we use in basic cancer research are predominantly of European or East Asian descent. And so we're not modeling all ancestry groups when we're studying cancer in the lab. And so this is a problem that definitely needs to be addressed. The second is that we identified many ancestry-associated genetic dependencies. At least initially, most of them were due to this artifact that we described, where there's mismatches in the guide targeting sequences. But excitingly, in our more recent analysis, we found that many of them are not, and so we're excited about really understanding the nature of these dependencies. And then finally, what we found is that there's an ancestry bias in all CRISPR-based experiments, and if you're not actively trying to correct for it, this bias disproportionately affects individuals of African descent compared to other ancestry groups. And so it really highlights this problem, I think, in how we're going through basic science research, and maybe we're just doing experiments, but not necessarily considering where forms of biases can come from. And so with that, I would just like to thank folks in the lab for their suggestions and their moral support. And in particular, I'd like to thank Aaron Coltonier, who's an RA in the lab, who's done a lot of this analysis. And in particular, he, with no help from me, single-handedly did a lot of the computation for the web tool that I presented. I'd also like to thank folks in GPP, folks in the pattern team, and also folks on the cancer data science team, and Alham Sadat for his suggestions as well. And happy to take questions if there's any.
Thank you so much, Sean, for that really interesting talk. I want to welcome anyone who's here, either in person or online, to raise any questions. I will start with one. I'm curious about this notion about avoiding guides that are targeting regions of really high diversity, and I wonder if, or avoiding them. And I wonder if at some point that becomes limiting, because of course there will be some regions that are of high diversity due to selection and historical phenomena. And I wonder if that is in practice an issue or if it's avoidable by careful design.
Yeah, so yeah, there are some instances where it's just unavoidable. There's so much variation in some genes, or the genes are so small that you can't avoid it. When Aaron designed the custom library that isn't ancestry biased, there were a few cases in which that was an issue, and you just have to pick guides that you think work best and like acknowledge the fact that that issue is there. But for a majority of the genes, it was as simple as just picking the guides 2 through 5 instead of 1 through 4, and avoiding the entire issue.
Whether or not it made sense to divide up the different ancestries, like as like African, East Asian, and so on and so forth, and if there was really a bias, because the group sizes are a little bit different. Like I think it's known that the genetic variability between like those of African descent is larger than genetic variability between those of European descent, so on and so forth. And maybe like whether there's a way to like measure that, and like maybe the bias is expected just because that group has more variance, like more intra-group variance. Whether you've thought about like trying to control for that, and I don't know, like measuring whether or not a bias still exists, or whether it's just dependent on like the size of the group.
Right. Yeah, I guess like in each, if we look at it on an individual level, like compare one, maybe this, I'm missing your question, but like if we compare like one European individual to one African individual, on average in that 1-for-1 comparison, the African individual is more affected than the European individual, independent of like group size.
Right.
But we can also like, we can substratify each ancestry group into subpopulation level ancestry groups as well.
Right. Yeah, that's what I was wondering. But yeah.
Yeah. I think that would be really interesting, but it's not. We haven't done it yet. But we have all the information to do that.
Question that's just appeared on the Q&A, and that is from Jason Kwan, which is, what efforts, if any, are currently ongoing to expand underrepresented ethnic cohorts with regard to cell models and in DepMap?
Yeah. So in DepMap, I don't know. There's this current project going on called HCMI, Human Cell Models Initiative, I think. And one goal of that is to increase genetic diversity in cancer cell lines. And so they specifically tried to, get samples to generate cell lines from institutions where maybe there would be more genetic diversity in the patient population. And that's still ongoing. I think it remains to see how much it will increase genetic diversity of the cancer cell lines we use.
Thank you for a wonderful talk today. I actually had a question before you step away. Is there biology to this diversity in CRISPR target sites, right? Like there's some consensus in the actual sequence of the sites, right? And so if you're looking to target, there's going to be some amount of similarity in the surrounding sequence where you're finding this variation but does it also map to any kind of appreciable biology in terms of differences in cancer susceptibility, or kind of, I know you were looking at how many of them were EQTLs, but is there sign that there's actually biology in this as well? Some of the SNPs that inhibit CRISPR.
Yeah, I'm sure there is. So yeah, I guess I can't think of any specific examples of where we've identified a SNP that's blocking CRISPR, but is also like just an interesting SNP in general. But I'd imagine there's like so many interesting SNPs that are altering protein function, and there's so many regions in the genome that we've designed guides to that there's got to be an intersection somewhere. And because I haven't found it necessarily in my analysis, it doesn't exist.
Certainly another reason, Sean, beyond the DepMap, when CRISPR experiments are underpowered, comparing 1 versus 1 or 2 versus 2. If you have ancestry-associated differences in those very small groups, you could bias an enormous fraction of the hits that emerge, even 50% or greater. So the depth map, I think you're exactly right that it's not a challenge, but for many of the screens that people perform in the community, it could be an enormous challenge.
Mhm.
Thank you. I learned a lot. I just wondered like what to make of those genes that are not even expressed, but nevertheless are the like dependency signals. Like, did you see any other explanations for those?
Yeah, so what I think it is just CRISPR is cutting at those loci and you get a little bit of cell death because you're getting that cutting. The guides just so happen to be targeting genes because that's how the library was designed, but you can imagine targeting intergenic regions or regions where you think like absolutely nothing is happening in the genome and you would still get the same sort of cutting effect. So I think it's really due to just the slight amount of toxicity or genotoxicity you get from introducing a single DNA break or 2 DNA breaks.
Sean, since you're being recorded, could I get you to speculate on the record? The cancer community thinks a lot about somatic variation. There are fractions of the cancer community that study predisposition that think a lot about germline variation, but I wouldn't say it's the epicenter of how precision medicine is implemented today. Do you think that even with its warts, these types of studies are forecasting a future in which all cancer genomicists need to think much more about the germline as they relate to therapeutic intervention? Or do you think that's still going to be at the fringes of our thinking and somatic variation will really be the epicenter of precision medicine?
Yeah, I mean, I think germline variation is awesome. So I'm obviously biased here. Yeah, I think both are important. I just think, we don't have enough maybe information about how germline variation influences cancer vulnerabilities to really say like what's the relative importance of each. I mean, certainly knowing that you have loss of function germline variant in BRCA is going to be important for patients who are trying to decide if they want a PARP inhibitor or not. And I'm betting that we're going to find other examples of this as well when we look. But whether those rise to the strength of saying that people with BRAF mutations are very sensitive to BRAF inhibitors, I'm not sure.